Hello and a very warm welcome, all distinguished colleagues and allergology enthusiasts joining this evening. It's a true pleasure to have you here with us for the first edition of our Fascination Allergologie webinar series. Tonight, we will collectively look beyond the traditional boundaries of daily practice and explore the exciting future that lies ahead for our specialty. Today's session is dedicated to a pivotal topic that is already reshaping clinical decision-making, research strategies, and ultimately patient care. We will take a closer look at the future of allergology, how artificial intelligence is transforming treatment. So, let us embark on this journey into the fascinating interface of allergology, data science, and precision medicine together. Before we delve deeper into our fascinating topic, a quick technical note. For those of you who haven't discovered it yet, we offer a translation option for this webinar. To activate it, simply click the language button at the top right corner of your screen and select your preferred language. Please be aware, our translation is highly advanced and generally very good, but like all technology, it's not perfect. If anything is unclear or if you have any questions about the translated content, please do not hesitate to ask in the chat. We want to ensure everyone can follow along comfortably. Now that everyone is set up with the translation options, I'd like to take a moment to introduce myself and the project Fascination Allergologie. My name is Christian Schwager, and I have dedicated the past 12 years to the field of allergology. My work has included contribution to the discovery and first description of several peanut allergens, and I am currently deeply involved in various trainings and educational programs, especially for young allergologists. It is a great pleasure to be a host and one of the moderators for today's session. And beyond my personal work, I am deeply committed to fostering exchange and education within our field. A prime example of this commitment is our webinar series Fascination Allergologie, which I'd like to briefly introduce. Launched in 2020, the German version of Fascination Allergologie has grown into a highly successful educational platform for allergologists, primarily across Germany, Austria, and Switzerland. We proudly hosted over 50 webinars, engaged more than 4,250 live participants, and had over 18,500 views on our YouTube channel. Our dedication has also been recognized twice with the Digital Health Heroes Award. And yet, beyond these impressive figures, our true driving force is our mission. We want to share the knowledge and passion for allergology to advance patient care. We want to offer exclusive insights into the personal and professional journeys of leading experts. And we want to serve as an open forum for hot topics and important issues, including women in medicine, work-life balance, and also career development. Today's webinar perfectly embodies this tradition as we explore one of the most exciting frontiers in modern allergology, the transformative role of artificial intelligence and biomarkers in diagnostics and treatment. And with this brief introduction and background established, let's now outline the concrete learning objectives for today's session. By the end of this webinar, you will be able to understand the current effects and future potential of AIT in the context of AI-driven insights, describe key approaches for biomarker discovery and their clinical relevance, and explain how AI is fundamentally changing allergology from diagnostics to personalized treatment. Now, let's review the structure of our evening, and as always, this webinar is divided into 3 distinct parts. First, We will commence with a comprehensive overview presentation. Here, our expert will guide us through the questions of how AI is transforming treatment in allergology. In the second part, we invite you to engage with an interactive patient case where you can test and apply your allergological knowledge. And finally, we dedicate time to your questions and your clinical experiences. Collegial exchange is a cornerstone of this webinar series. So please seize this opportunity to ask what matters most to you, whether it's about today's topic, career development, or specific patient cases. And direct exchange with you matters really to us. We warmly welcome your questions, comments, and personal experiences. Please feel free to share your observations and to submit your questions directly to us and your expertise. And to the expert, please use the Q&A panel on the right-hand side of your screen. Our expert will share his knowledge and practical experience addressing your contributions throughout the webinar. And if the Q&A panel is not immediately visible, you can easily open it. Simply click on the chat icon at the bottom right corner of your webinar window. And this webinar is CME accredited, so please note that the continuous attendance of the webinar is required for getting the CME credits, regardless of which device you are using to watch. And furthermore, I want to briefly highlight the post-webinar evaluation. Your feedback is incredibly valuable for us. So please share what you found beneficial and what could be improved, along with any suggestions for future webinar topics. You will also have the opportunity to rate the AI translation, which is crucial for us to continuously enhance our services. And now that we have covered the organizational aspects and given you an overview of the structure of this evening, it is my great pleasure to introduce our speaker for today's webinar in more detail, Professor Mohamed Shamji. He will take us on a journey into the world of artificial intelligence in allergology. So Professor Shamji is a trained clinical scientist in immunology and completed his PhD in clinical medicine at Imperial College London. He is now Professor of Immunology and Allergy at Imperial College London, head of the Immunomodulation Group, and Director of Translational Allergy Research at the Frankland and K Allergy Center. His research focuses on type 2 inflammation, immune tolerance, and allergic diseases such as rhinitis, asthma, and food allergy, using big data, AI, and machine learning to identify mechanisms and biomarkers of allergen-specific immunotherapy. In addition, he is EAACI President-elect, EAACI Secretary-General, and Chair of the EAACI AI in Innovative Trial Design and Precision Immunology Task Force. And however, we know that someone who works so hard for allergology and for patient needs also some time to relax. So we have a question for you about Professor Shamji. A short poll will now appear on your screen, and we would like to ask you, what are Professor Shamji's hobbies? So please choose one of the following options. Is it cooking and trying new recipes at home? Or is it playing the piano and listening to music? Or is it maybe C? Is it golfing, playing golf, and going cycling? So please choose one of these answers, and to get it up a little bit, so we will speed it up, and I would like to show you the countdown, which will give you the last 5 seconds. All right, then let's have a look at the answers. We will maximize, and you can see that about 30% are for cooking, answer R. Almost 30% are for playing the piano, and 32% playing golf and going cycling. And the right answer to this question is, as you can see it here, golf and cycling. And now it's my distinct pleasure to introduce our esteemed expert for this evening, Professor Mohamed Shamji from London. Welcome, Professor Shamji.
Thank you, Christian. Delighted to be on this podcast, and thank you so much for the invitation. I'm very much looking forward to this session.
Yeah, and now let me introduce you the two colleagues who will confidently guide us through tonight's program. From Copenhagen, it's the charming and eloquent Professor Anne-Marie Choos, and from Nuremberg, our multi-talented colleague Sebastian Sorich. A warm welcome to both of you.
Hey there.
Thank you. Thank you so much. Hi, Mo. It's such a pleasure to have you with us today. We're very, very excited to hear your talk. We'll start out with one question, which is obvious by the title of this seminar or webinar. So what fascinates you about allergology?
Well, thank you, Anne-Marie. Delighted to be here. My fascination with allergy began with my training in immunology. Clearly, I developed affinity towards during my training in an allergy diagnostic lab and research lab, and actually I realized that amongst other the challenges that we see with various different immunological diseases, at least we know the allergen with allergy, and we know the global impact of allergic disease. And also we do know the allergen, so we can think about prevention, we can think about treatment, and also managing patients. So I thought the best thing to do is to, go into the field.
Yeah, that's so nice to hear. Going to the field, and there's still a lot of work right ahead of us as well.
Indeed.
Today, we want to talk about AI in allergology, and I, in the background, already started a short poll for our audience and asked them how familiar do they feel with the use of AI in clinical allergy practice. And most of them are curious but unsure where to start. The other ones are not familiar at all, and just a few are very comfortable, already use AI-based tools or somewhat familiar, and some are even slightly skeptical towards AI in medicine. So we're very much looking forward to your presentation. Maybe you can clear up some stuff and tell us more about how we can use AI in allergology in the future. This stage is yours.
Well, Sebastian, I'm not really surprised with the poll. I think with AI, there's always this conception of understanding AI because it seems very far off, but actually it is here and we are using it. So these are my disclosures for my talk today, and I'd like to you've kindly asked me to talk about the future of allergology and how artificial intelligence is transforming treatment. And I think we're really seeing a huge change where AI is beginning or increasingly being used in our daily lives, but also in our specialty, particularly allergy and immunology. And I hope I'll give you a little bit of a background on how we're seeing this developing and shaping what we do, particularly to improve patient care. Well, firstly, what I'd like to do is to, we talk about AI all the time, but do we really understand AI? What is artificial intelligence? And I think it's important to really appreciate the definition and then how this, how we can use it and how can we feel comfortable even talking about it and how we can use it. So artificial intelligence is a computer science field focused on creating systems that can perform tasks requiring human intelligence. So I think that's really important. And the word AI was coined by Professor John McCarthy. He was an emeritus professor at the Stanford University, and he really proposed and co-authored the proposal for the Dartmouth workshop, which coined the term AI in 1956. And he really described that computer can perform tasks based on experience and set rules. And that was the first evidence, or the first evidence we know in terms of AI that was coined in 1956 by John McCarthy. And I think when we consider the use of AI today, we see this the utility of AI in chat boxes, in sat navs. We do see it in automated hoovers and gaming and so on. But I think it's very important to really differentiate what is really artificial intelligence compared to what is the task that a machine is performing. And those tasks are actually led by machine learning algorithms. So algorithms support the task that you ask a machine to do, for example. But really, the difference is very clear. That when we talk about AI, are we talking about artificial intelligence, or are we really considering machine learning algorithms? And as you'll see, as we move towards my talk, we tend to use mostly machine learning algorithms to perform tasks that we would like to support us in some of the activities in our clinic or in research. So I had the privilege to be part of the practical allergy, which was convened by the American Academy leadership and the European Academy leadership, Paul William and Maria Torres, and I had the privilege to be part of the leadership and a member from the European Academy to really try to understand and how can we really bring and think about AI in a context of allergy and immunology. So it was a true pleasure to be part of this initiative. And the first thing we did is we went back and we thought, okay, well, let's just try to understand, the development of AI and its utility over the years. And it became very apparent that when we think about, the first evidence of computational science, started with the abacus. And then we had transistors, which can perform complex tasks, complex computational, and can store data, and thinking about AI and its utility. And then throughout the timeline, what we saw in the 1970s particularly, there was lack of funding to support development of AI, which then picked up very nicely with development of LLMs. We are now seeing very much virtual virtual headsets. We are seeing ChatGPT, and now we are increasingly using AI tools like ChatGPT in our daily lives. And I think it's not just us professionals, but also patients are using it. So we've seen a trajectory of change over time and development of AI and how that's progressed. But is it again artificial intelligence, or is it just development of machine learning algorithms that are supporting some of the tasks that we would like to achieve? But it's very important to really think about the pillars and the ethics of artificial intelligence. I think it's very important to appreciate that there are many elements of bias in ethics, and if AI is not performed properly, it can be used inappropriately at several stages. Now, we need to think that when we talk about AI, at least from this industry standards perspective, it needs to be fair and regulated. It needs to be really interpreted. It's got to be interpretable and explainable as well at the same time, because it's not just a black box. You don't just add your data and something comes out, but you need to really understand really how well and what's really happening. I think the other element is got to be auditable. It needs to be transparent. And also one needs to think about how one can use AI responsibly, because you can have access to the data, and then you run these machine learning algorithms that allows you to perform tasks and analyze your data, but you need to be very careful how you use it, and one has to be really responsible and transparent about how AI can be used. But when we really consider the use of AI in clinical practice, we know, for example, AI has been used in the context of electronic health records, recording of conversation between a physician and a patient. And while this is happening, it captures the information, it sends the information to the physician's computer and summarizing it, and somewhat also capturing some of the key very key points that the physician may then be able to make a decision in terms of prescription. So we're seeing increasingly, at least in the US, how AI tools are being increasingly used during consultation. But it's important, again, to just stress that it's extremely important to think about the ethics of AI and how this is important in a way when how to use the data, the security of the data, and also constant check of the data once it's been captured and how it's going to be used. It's also important that to note that our patients are becoming very savvy. They use ChatGPT. They're able to upload their results, laboratory results, and they almost are able to self-diagnose. And how do we manage that in our clinical practice? It's extremely important to think about that, and how particularly when you have a patient who come and sit in front of you and is telling you about what ChatGPT has diagnosed, or what the results meant, and so we are increasingly dealing with very savvy patients, but that's okay as long as we understand how to manage. And I think education really plays an important role here, particularly in a context of educating the physician, but also appreciating that what we have to do in a context of ethics, and how it is extremely important to deal with this in the clinic, and therefore moving on to the future of AI. Where's the future? Well, we know that there are elements in terms of digital twins. How can AI support diagnosis, patient management, and so on? But I think it's important to really think about how we use this AI responsibly. So moving on to the other aspect of AI, as I mentioned, AI is moving into the clinical space, but what about in terms of allergy research, which is mostly the area that I am in, and my work, my professional work is. So we know that when we think about allergy research, we think about a novel therapeutic, we think about design of clinical trials. AI can be very, very helpful in terms of consideration of site selection, ethnicity. So if you have all the information that you need, you can select the site that will provide you with the best patients that you want to run in your trials. It can support trial enrichment. And also in the future, what we think we can also see the role of AI in a context of digital twin. But as you move from design to recruitment to then monitoring, of course, we're seeing increasingly wearables that are using AI. We're seeing how we can integrate the environment exposure into the mix here, and how that would relate to the clinical response. And then, of course, we really have to think about the real-time data analysis and how this can move forward in a very unbiased way, and more of a supportive way of running studies mostly efficiently. So there are some benefits and there are some challenges, particularly in a context of AI in research. You have adaptive trial design. You can really think about how best one can use and resourcefully having a large data set, and how one can use AI to support clinical development. But I think it's also very important to really have a clear balance of transparency and bias, and also understand the AI models that are being used to identify certain patients for, let's say, trial arrangement, and so on. So it's very important, and we're seeing this increasingly coming into the allergy research space, and particularly when we also think about healthcare speciality. And I think it's very important to really appreciate that AI is not just for allergy and immunology, which is the topic for today, but I think most importantly, what we're seeing is that it's increasingly being used in cardiology, in dermatology, in pathology, and radiology, as well as surgery. So AI is here to stay, but we need to make sure that we are using it safely, transparently, and also with good ethics in mind. But most importantly, the topic for my talk in a context of AI in allergy and immunology. So what we've really noticed is that AI is increasingly being used in respiratory disease such as asthma, particularly to phenotype or endotype patients. You have machine learning algorithms that are supporting that. It can support a development of trajectory of exacerbation, support identification of risk cause, and also patient health, track patient health, particularly when, with wearables. In food allergy, what we're increasingly seeing is how AI and certain machine learning algorithms are able to support or try to predict reaction to oral food challenges, and whether it's just looking at the exposure to the allergen and predicting allergen and doses and predicting oral food allergens, or whether it's a combination with biomarkers. And there are certain algorithms that are, that can support whether one can be in the more sensitive group here. In the context of dermatology, we're seeing AI being used more often as well. We're seeing drug allergy as well, but also innate error of immunity. Particularly, we're seeing that the AI, the algorithms are pulling out a lot more of the patients for diagnostic purpose. But the topic for me is actually considering is allergen immunotherapy, because when we think about allergen immunotherapy, and certainly in a diagnostic workup, we collect a lot of data. We have skin prick test. We're able to identify the allergen and the major and minor allergen particularly, so we can do CRD. But of course, we have the clinical data, which is very, very important. And beyond that, if one wants to understand underpinning mechanism of disease, you can perform multi-omic analysis and have a big, rich data set. And that can then support with certain machine learning algorithms to identify, diagnostic biomarkers or improve biomarker diagnostic accuracy, or predict long-term efficacy, and also identify biomarkers of response or no response. Well, when we really think about allergen immunotherapy, we know that allergen immunotherapy is a highly effective treatment for IgE-mediated disease, with or without asthma. It is associated with reduction in symptoms, the need of rescue medications, and improvement of quality of life. Most importantly, it is a disease-modifying treatment. Hence, it confers long-term clinical benefit and sustain immunological tolerance. And when we really try to understand the underpinning mechanisms of allergic disease and immunotherapy, we know firstly, and I'll just summarize this, what we know is we have a disruption of the epithelium, and we have a lot of alarmin that are able to activate innate immune cells that can support development of Th2 cells and Th2A cells. And these Th2 cells can support can help B cells to produce IgE. And then you have sensitization of mast cells in the local tissues as well as in peripheral blood, mast cells and basophils, and subsequent exposure results in the symptom that we see, which is sneezing, itching, rhinorrhea, the early phase responses. And then recruitment of the T cells into local tissue results in the late allergic response. But with immunotherapy, what we really see here is the resolution of the integrity of the epithelium. We know that the dendritic cells that are captured that are able to still capture allergens, they're somewhat modulated, they're less inflammatory, they support the differentiation of Th1 cells. This is immune deviation, induction of regulatory T cells, and these Tregs can support Breg induction, and you have induction of antibodies like IgG, IgG4, and I think that's really important to bear in mind. So, but one of the key elements here is that when we consider allergen-specific immunotherapy and its effect to treatment, there is still a challenge where we don't really have a clear biomarker of response. And although we know that we need humoral biomarkers, we can measure humoral responses, we can measure cellular responses, the changes in cellular responses, but there isn't a clear one biomarker that may be able to tell us that a patient is likely to respond or no response. And I think it's very important to really think about how we move forward. But just this is a couple of examples, and this is a study by Stephen Durham. This is one of the seminal study where he demonstrated that if you give immunotherapy for 3 years, you are able to induce long-term sustained efficacy for another 3 years. This was a randomized double-blind placebo-controlled trial. You had 47 patients here at the age of 3 at 3 years of treatment. If you look here, this is the pollen season. These are the symptom scores, rescue medication scores, and the VAS. And at year 3, the orange line is below the red line, highlighting that those who were on treatment had lower symptoms, need a rescue medication, and also VAS score compared to the placebo, which are the red line. And after 3 years, the treatment group was randomized to either be on sustained maintenance or withdrawal of treatment, and they were followed up for 3 years. But he also recruited an untreated grass pollen allergic control group that he followed over the course of 3 years, and you can still see here that either the red and the blue lines over the course of 3 years, they remain low when he looked at symptom scores, rescue medication scores, and also VAS scores. So clearly a clear definition of and demonstration of long-term persistence, long-term clinical benefit after 3 years of treatment. And we've seen this as well with the SLIPT grass pollen sublingual immunotherapy, where this is a large randomized double-blind placebo-controlled trial. Patient received treatment over the course of 3 years, and this is the data for the 3 season, and there was a clear sustained reduction at year 1, in the first season, second and third, consistent reduction in symptoms compared to placebo, and again a sustained response following discontinuation of treatment and the sustained clinical benefit here. And this is the both 2 studies that I showed earlier with grass pollen immunotherapy, SCIT and SLIT, and this has nicely also elegantly been demonstrated in a context of house dust mite. This was a study, a randomized double-blind placebo-controlled trial, a huge study with a dose-ranging concentration of the two doses in the active group and a placebo. And in terms of the clinical benefits, patients were followed, and a 1-week diary was collected over the course of 10 months, and also 15 days before treatment, and also the daily diary were collected over the last 2 months. And the key, the aim of the study was to reduce symptoms of rhinitis and need of rescue medication score, a huge population where house dust mite allergics with mild asthma, and a clear primary endpoint that was very much evaluated here. But it was very clear that throughout the year there was a year-round effect with early onset observed at 14 weeks in terms of the clinical benefit and using the two doses, and also the number of days per year with allergic rhinitis exacerbation characterized by moderate to severe symptoms despite medication was reduced from 40 to 19 days. So the stuff works, whether you give it for grass or whether you give it for house dust mite. And we also know that for sure that the SCIT or SLIT are both equally effective. But the challenge for us has always been, what are the key biomarkers for immunotherapy when you have a patient in front of you in your clinic and you want to really identify, is this patient likely to receive respond to SLIT or SCIT? Are they going to respond within the next 3 months or 12 months or 24 months? Would they be the one who will have no symptoms after discontinuation after 3 years of treatment? And I think it's very challenging. We are not there yet, despite understanding the several mechanisms of immunotherapy. And the key for us is really to think about in the context where we know the mechanism, but we need to really conduct the relevant studies. So, for example, if you give immunotherapy over the course of 36 months, where we know you can induce sustained clinical benefit, whether it's for SCIT or SLIT, you need to think about relevant biomarkers that will tell us response, no response, or even placebo effect or safety in clinical trials, but also in the clinic particularly. Can we identify biomarkers of desensitization? Can we really identify biomarkers of efficacy? Should efficacy, other biomarkers of efficacy at 12 months vary compared to 24 months and also 36 months when we are seeing tolerance? So, and with that, it's a lot, we need to conduct several mechanistic studies, and then really think about the tools we can use. And I'd like to share with you some of that. So this is a randomized double-blind placebo-controlled trial of SCIT and SLIT trial conducted by Stephen Durham at the Royal Brompton Hospital. I had the privilege to work with Steve and be part of this study, particularly on this study. And this was a randomized double-blind placebo control where patient received SCIT, the red line, SLIT, the green line, and the blue line, which was placebo. Patient received treatment over the course of 2 years, and then they were followed for 1 year. We had access to several clinical outcome measures of efficacy were evaluated outside the pollen season, particularly looking at TNSS following intranasal allergen challenge at year 1, year 2, but also at year 3. We also looked at quality of life of patients as well, and we had the opportunity to conduct several mechanistic analysis to look at T-cell responses, B-cell responses, antibody responses, and we had, and this was a really great study to be able to really evaluate not just mechanisms of tolerance, but also biomarkers of tolerance. And we were very lucky to be funded by the Immune Tolerance Network. And this is really the clinical data. These patients following intranasal allergen challenge at baseline, you can see all the 3 groups are matching. Year 1, SCIT and SLIT was effective, remained effective at year 2. We lost that effect at year 3. There was a placebo effect. And we also looked at peak nasal inspiratory flow, which was improved at year 1, year 2. We lost that effect at year 3, and we also had less of a placebo effect. So in a sense, although it may have been very disappointing that we lost that clinical effect at year 3, but perhaps having access to having done several mechanistic analysis and data, this was perhaps the best study where we could have responders at year 1 and year 2, but also have non-responders at year 3. And I think access to data is extremely important. And if you pose a question and you have the right study to address that, it really helps in a sense, particularly in the context of biomarkers. So just to give you an insight of the type of analysis we did, we had over 70 features of immunological and fundamental and clinical parameters that were measured in the GRASS study. We had access to blocking antibody data, IgG4 analysis, to components, to GRASS pollen components following treatment at year 1, year 2, year 3, also during the pollen seasons. We had access to IgE data, IgE responses to the components, IgA antibody, IgA1, IgA2 to the components. We had access to patient demographics. We collected, since we performed the intranasal allergen challenge, we were able to collect nasal fluids and measure proteomic analysis looking at T2 cytokines. We performed basophil activation analysis. We did a lot of T-cell work there. So I think it was really, really great that we were able to work on this. And having access to this data, the question we pose is, we had a huge data set, but what can we do with it, right? Can we really identify the signatures of biomarkers that will tell us, response and no response at year 1, year 2, year 3, and how can we do that? So I have 2 great bioinformaticians in my group, and they really are fantastic, and it was a pleasure to work with them. And there were 2 types of questions we had asked. So the first thing, so this was a 2-step analysis approach, which was used in terms of identifying biomarker of response and no response. And one can do one single variable analysis. So you have, let's say, you look at IgG4 at all the time points at 2 different groups, and then you correlate that with clinical response, and then you see how that goes. Or you just take all the data, all the big data into a pot, and you run your machine learning algorithms and see what comes out. Are you able to pull clusters of patients who are responding or not responding to treatment? And if you do, what are the key biomarkers that will differentiate them? And that's really what we did. So we had step 1 approach, which was single variable, and step 2 was to really look at to perform some integrate all the features of the data and run some machine learning algorithms to really evaluate whether we can pull out responders and non-responders. And here you can see that you have a list of several machine learning algorithms that one can use. And here the key point was we used the data to train. 80% of data was used to train, and 20% was actually used to test. So if you were to do signal variable analysis, which is most of the time what we would do, is that you can achieve the following. For example, here you have specific IgE, ratio of G4 to E to FLIPI6, and you can identify, for example, in placebo, there is no response. In SLIT, there is some form of response, and in SCID, there is a much enhanced response. Or you can have another variable here, again, a ratio G4 to E, for FLIPI12, and what you have here is you've got no change at all. And if you look at this and you literally just plot a heat map, and what you'll find is that there are certain biomarkers that are coming up so they're great at stratifying active treatment or no treatment. So for example, in SLIT, you have year 1, year 2, and year 3. These biomarkers are blue, so they're you can see they've gone up, and somewhat they are. We're seeing it in SLIT and in SCID, and it's not differentiating much to patients, but it's somewhat different than placebo. What you can also do, you can look at a second series of biomarkers that are not coming up here in placebo or SCID, but they're somewhat flagging up at year 1 and year 2. And really, that's the key. And we can also look at this data analysis, and the key was to think about whether we can identify biomarkers of response and whether they can stratify responders. So just looking at the data where we have responders and non-responders, responders, partial responders, and non-responders, year 1, year 2, and year 3. First of all, you can see that when you have SCID and SLIT, once the placebo effects wanes down from year 1 to year 2, it stays the same. And most importantly, that was the key element. And SCID and SLIT was more or less the same in this context. But also, you can also identify biomarkers that are somewhat the same or not. So here we are looking at G4 to E ratio for FLIPI6, and you can see that in SCID they are coming up, but in SLIT they're somewhat very different. So interesting to, they can differentiate at least the response to SLIT, but not differentiate the difference between SCID and SLIT. And if you then look at year 2, for example, when you want to look at the analysis, just looking at responder analysis, what you're able to see is that there are certain biomarkers here on the left. And if you look at the dotted lines, that in SCID, they're not flagging up at all, but in SLIT at year 2, they're appearing somewhat blue and not orange and yellow. And there are certain other biomarkers at year 3, for example, they're coming up mostly in SCID, but not in SLIT. So it is somewhat challenging in a way, because we're identifying signature, but what does it all mean? And therefore, the next thing to do was to really think about, okay, we have access to this data, we will put it all together, and we will really think about how best we can use that and use the relevant algorithms to give us some form of information. And what we found, certainly, and I'm just summarizing this, there were several features that were associated with biomarker, with response to treatment. And certainly the T cell data, Th2 cells were very much associated. So low Th2 cells at baseline were associated with better effect size of treatment. What we were also able to see that you can look at several antibody responses and how they had a role in somewhat identifying response to treatment. And the key element for us is that however you looked at the data, there were some signatures that were coming up, but they were associated with pathways that we know. So that was very important because it's explainable AI. We're able to really identify that Th2 cells, Th2A cells play an important role, and the fact that the proportion at baseline are very much associated with response to treatment. And other parameters also, for example, nasal IL-4 was also very much associated. And again, T2 inflammation plays an important role on how how we see response to treatment. That was very interesting. And we're still currently doing a lot more analysis. But when we try to do these sort of algorithm analysis and try to predict non-responders at year 3, the algorithm somewhat, struggle. So then we pose the question, could we have a different signature for year 1, year 2, and year 3? And this is really what we did. So we run the algorithms and we looked at accuracy of the algorithms and how best we could, and which one would be best to use in a context of sensitivity and specificity of signatures of biomarkers that will be very relevant for identification of relevant biomarkers for year 1, year 2, and year 3. And it was really interesting that at year 1, there was a key signature of biomarkers that came up that were associated with response to treatment. At year 2 particularly, we also identified signatures at year 2, but they were very different. They're somewhat different than year 1, and also at year 3 as well. What this tells us that there is somewhat of a temporal change in the relevance of biomarkers that are associated with clinical response to treatment. So the biomarkers at year 1 are not necessarily the same biomarkers at year 2 or year 3. And we wouldn't be able to do these analyses if we hadn't used, some form of AI or machine learning algorithm to really tell us that. So again, what I usually say is, efficacy of immunotherapy is associated with immune modulation at different layers, cellular responses, humoral responses. It's like playing a piano. You play a piano and you have once you have a very nice tune, everything is very smooth. But, you have to really be able to play the right keys to modulate and to really bring this wonderful sound. And actually what we're saying is that in terms of identification of biomarkers to response, we need a composite approach where we need several different biomarkers of cellular, a composite of cellular and functional readout that may support efficacy of treatment. But what's really important is that, how do we move forward? What is the future? What is the future of biomarker discovery? So we know that you have a cohort of patients. Our patients are very diverse, but what we need to really think is we need to really consider how we phenotype them, how we are able to perform humoral response analysis, how we're able to use transcriptomic analysis to understand the disease, how we're or to evaluate immune response, how we're able to think about looking at proteomic analysis, but also serology, and having access to several components of immune response, and really using data science to then come up with signatures of biomarkers would probably tell us about, the high, the low, and the non-responders. And actually, even better to identify the non-responders so we can do something about it. And with that, I'd really like to thank my the team that worked on this data set particularly, but also the privilege I had with working with the American Academy leadership and the Yaki Academy.
Thank you very much for this nice presentation, Mo. I really enjoyed it, and thanks for the big overview about the applications and the research that's already been done about AI and AIT. Once again, I want to encourage our audience to ask questions in the chat on the right-hand side. But I think Anne-Marie is going to start with the first question that we have.
Yes, thank you, Mo, for a very fascinating talk on this very, very hot topic. Many clinicians still feel that AI is sort of a buzzword. From your perspective, what are the most realistic and immediate applications of AI in allergen immunotherapy today? And also, for the clinician sitting with the patient in front of them, what are the most realistic applications that we're going to be using in clinic?
I think certainly the way that AI tools are able to capture consultation with a physician, I think that's really important in a way, because you're able to capture your conversation with the patients. You're able to discuss whether the patient is going to be whether you'd like, the key before you onboard them onto immunotherapy, you'll have this conversation with them in terms of their diagnostic results. You will be able to discuss with them in a context of how long it takes to induce efficacy. We know with scatolostates it's very quick, 3 to 6 months, and then you have efficacy and treatment, and of course you have also adherence to treatment. And I think this capturing of information and also being able to what I see in the future, I must stress that is perhaps if this were to be geared towards immunotherapy, there will be the cue that the notes that's collected with the proposition of discussion and particularly how best to manage the patients is where we'll be seeing this going forward for sure.
Anne-Marie already mentioned that AI is kind of a buzzword, and that's also something that we see in research, and we try to apply AI on many applications where it's actually not really necessary, and classical statistic methods would perform just as well. That's why I really like that you also showed that you actually approached it from a single variable approach and looked, well, what is actually the benefit of machine learning? And maybe you could just summarize that once again. Where would you prefer classical statistical methods, and where do you see machine learning actually performs better?
I think that it's important. That's a very important question you posed there. We need to be realistic. We need to think that the statistical approach is extremely important, and it's the sort of the gold standard at the moment. And I think we're all using that, and it will be the gold standard for a while. I think AI, the strength of AI is when you have a large data set, huge data set, where you want to perform unsupervised or supervised cluster analysis. You want to be able to do something completely unbiasedly and then understand what's happening within the data. And I think that's somewhat the strength of the AI tools that we are seeing coming in. So I think they can be very complementary for sure. And I think that's really the most important point. So what you can't address, where you find that you want to be a little bit more exploratory, that's where we can think of using AI. And I think what's really also important is that is having access to great bioinformaticians is also very important, because the field is moving at such fast pace that the algorithms are changing, because access to, it's just really fast paced, and some of the algorithms are also expiring fairly quickly. So taking it, having a great conversation, and it's really important to work with a bioinformatician so that they understand what we're trying to do, what we are trying to achieve. The data I showed you was it's very nice and still very challenging in the context of allergen immunotherapy. We've worked with some of my bioinformaticians have worked with another topic in collaboration in terms of diagnostics for hereditary angioedema and endotypes of patients, where it's extremely clear that you can even have cohorts from two different cohorts completely world apart, and the algorithm just works so nicely, and they come up and say, oh, this is working really well, but the other data is very challenging. So it is what it is, but I think it's a developing field, and most importantly, I think having access to bioinformaticians who are also interested in some form of biology or clinical aspect of medicine is also very important here.
Maybe just to follow up at that point, when you talk to bioinformaticians, do they say that the models or the progress of the models is currently holding us back, or that it's actually structured relevant data that is not acquired right now in a good section? So what's the problem right now?
Yes, Sebastian, very good question here. I think there's a bit of both, really. The first one is what you put in is what you get out. So it's extremely important that the studies moving ahead. What we've learned is when we are conducting study, we firstly discuss with the statistician in terms of sample size and everything else, because that's very important, very crucial, gold standard stuff. And then the second thing is to discuss with our bioinformatician so that we have that template, because it's about depth and breadth of the data, and what we put in is what we get out. So it's extremely important to think about that. And then, of course, you have a certain limitation of the algorithm. Sometimes you have to develop new codes. and that's what they are there for. And then you also have IP on those codes. But it's actually really fun. It's actually quite fun because we always sit on data. We publish a lot of data, and once it's published, they're usually sitting somewhere, and actually having access to all of that together, and particularly collaboratively, is extremely important, so that we can think of what else can we do in the context of understanding the data that we capture. I think now we can elevate beyond the statistical analysis what else the data is telling us, and I think that's the beauty that still to be unraveled.
Yes, I was also very interested in your talk about multi-omics and multi-omic workflows and integrating that into AI. I noticed, or maybe I missed it, but you didn't have much about the gut microbiome or the oral microbiome. Do you think that plays a role as well in predicting who will do well with the immunotherapy?
Yeah, absolutely. I think the data we presented was on the basis of what we captured, and of course, multi-omic analysis is extremely expensive, as you can imagine. But actually, what's really important is to think about, the question is it the data that we collect and that's a limitation or not? And I think you brought in a very important point, Anne-Marie, that, we have to think about the metagenomic analysis, the microbiome. There are some studies that have shown that there is a link in terms of response, that, better response with some of the key signatures in the microbiome, and that's just to do with inflammation. And I think we have yet to do those kind of studies where we collect everything at the same time and then we put it all together. So we have this rich clinical data set. We have the functional stuff, which is extremely important, because one thing we have to be very, very clear and important. It's important that we don't live in our virtual world. The omic analysis is great, but it's virtual. You can do the analysis, you can get the clusters, we can come up with some certain signatures or some indication of response, no response, but it's a virtual. We have to validate that. We have to go back and do a prospective study and demonstrate that those signatures are truly valuable in terms of clinical management.
Agreed.
Mm-hmm.
You mentioned before that international collaboration will also be, or will also play a major role in training machine learning models in the future. Why is it so important that there is going to be international collaboration, also looking at different ethnical backgrounds to generate realistic models that do well in prediction?
Yeah. As you probably know that a lot of the studies are done in Caucasians, right? For drug development and for a lot of the drug that's available, and I'm sure it's the same also for immunotherapy. And I know that LK and also other studies, other companies are also doing studies outside Europe, and that's great because we can have access. We can understand how immunotherapy works in other ethnicity, but it's very important to really think about the ethics of being able to have a pool of patients, but from all the diverse group, and really think about how best that works together. The other aspect is that, for example, we know very well CRS in Asia is very different endotypes compared to the CRS in Europe. So having access to international data sets, collaboration, only help us understand the disease better across. And actually, we are such a global community today. Whether if you're in the West, you have some Asians, and whether you're in the East, you have some Caucasians there, and Western. We're such a global population, and you don't want to be limited to the fact that the treatment only works to a certain ethnicity. So I think it's very important to really bring that into the mix. And actually, and collaboration is extremely important, and also transparency. So if you are a center of excellence, and you have access to patients, and you have information out there, and that really supports. So when one wants to do a trial, they can consider very nicely that, this center particularly has a great cohort, and in terms of the key parameters that one would want to have in their studies. So it certainly helps drug development, but also can support other type of studies, post real world evidence studies and so on. I think there's somewhat limited evidence with real world evidence studies and how that can be used. I think it's also important to understand the challenges, but I think it's still, the field is still in its infancy. I mean, it's fast developing. It's very independent on different sectors and different so on. But it's coming, and it's going to be here. It's going to be here. And we might as well learn about it a little bit more. I would just, my advice would be, if you're thinking about AI and using a machine learning algorithm, it's just a tool. So it's basically, you have a data, you use a tool, and it works on your data, and it tells you something. It's the same way as we would do, we have two data set, we want to do a p-value, we want to get a p-value between the two groups. It's a tool that's, that we can use on certain websites or certain software, but this is slightly more sophisticated in a way that it does something and it tells you what it is. But whatever you get from the analysis, the most important thing is that you need to be able to explain how that relates to your original data. And I think explainability, causality and explainability is the most important thing.
Thanks for pointing that out again, and also about the international data set. That's very important. Maybe we can continue this discussion after your case that you also prepared for us. I don't want to steal any of your time, so maybe we continue with your case first, and also see if there are any questions from the audience afterwards.
Sure. Okay. So what I have for you today, I have a 20-year-old male with seasonal symptoms, 5-year history of debilitating seasonal symptoms, usually coming from May to July, very, very severe and debilitating symptoms. And presenting symptoms particularly are severe nasal congestion with profuse rhinorrhea, intense sneezing, bilateral itching and watery eyes, palatal itching, and significant sleep disruption. The impact, particularly for this patient, is in terms of quality of life, 10 to 15 days off during the annually, particularly. The patient feels really sick 10 to 15 days annually during the peak season, unable to participate in outdoor activities, particularly between May and July, and considering a career change due to symptom severity. The patient has a past medical history of mild childhood eczema, no asthma, particularly in this patient, and a family history has revealed that the father has hay fever. The current management is oral antihistamine, which provides minimal relief, has been on intranasal corticosteroid with very modest improvement, and has had to use, in fact, oral corticosteroid courses 2 times in the last season for severe exacerbation. And in terms of the symptoms, you have severe disease and poorly controlled despite pharmacotherapy. And the diagnosis for these patients involved, appreciating and understanding clinical history, performing allergy testing, in vivo and in vitro testing, and also particularly we know that if you were in some patients, one needs to also measure spirometry if needed.
Yeah, you can read out the question more if you like to, and also the answers, and then we will start the countdown later on.
Sure, thanks. So the questions I have for you today, and let's see how you do, all do, and I'm sure you do superbly. What is the most appropriate diagnostic workup for this patient to confirm allergic rhinitis and identify the culprit allergen here, the key allergen that is driving symptom? So A, is it nasal endoscopy, CT sinuses, and amphitrial of immunotherapy, empiric trial of immunotherapy, skin prick testing to allergen panel, including grasses, tree weed, dust mite and molds, baseline spirometry, serum total IgE and chest X-ray, nasal cytology and allergen challenge testing only, and MRI brain and referral to the ENT experts for surgical evaluation.
Okay, so you have heard all your options, and I would like to encourage you to vote for A, B, C, D, or even E, if you think that the answer is correct. I will give you now your final 5 seconds, and then we will have a look at the results. Okay, here are the results, and I think you can see that most of our participants are voting for B. What do you think?
Great, and well done. I think that's that was the answer indeed. So really well done for picking that up. So the next question is related to the indication for allergen immunotherapy. As we've identified, our patient is sensitized to grasses, and we need to really consider whether if the patient is appropriate for immunotherapy. And we know that when we think about indication of immunotherapy, we need to confirm IgE mediated disease, which is what we've achieved. The patient has inadequate symptom control, which is what we've identified through our clinical history, and has a significant impaired quality of life, as the patient is unable to sleep at night, particularly. And of course, there are several other factors in terms of the desire of patient to receive treatment that would really work. And really, these are the key criteria for indication of allergen immunotherapy. So, having discussed that, and given this patient's confirmed grasp on allergy with severe symptoms despite optimal pharmacotherapy, which immunotherapy approach would be most appropriate? A, skid with mixed allergen extract, so grasses, tree, dust mite, and molds. Slit, drops with grass pollen mixture initiated during the peak pollen season. Either skid or slit tablets or drops with grass pollen extract initiated pre-seasonally or year-round. Oral immunotherapy with grass pollen extract mixed in food, or intralymphatic immunotherapy here as a first-line treatment. So what do you think is the right answer? I'll hand over back to Christian.
Yeah, thank you very much. I think this is a rather difficult one, or maybe not. We will see what the participants will choose. I can see there are incoming answers to this question, and I think that a lot of them are right, so really correct. So to speed it up a little bit, I will start the countdown timer. Then you have 5 seconds remaining for answering the question. All right, so we have a look at the results, and you can see that most of the participants have voted for C, meaning either skid or slit with grass pollen extract. What do you think, Mo?
Well, fantastic. I think, and the answer is, of course, C here, and the patient is in the right hand with this audience, for sure. So moving ahead, when we consider really the clinical benefit of allergen immunotherapy beyond symptom control, there is an element of, clinical effects, how soon we see this response. We know that immunotherapy has effect on prevention, but also long-term sustained responses. And I think that's really important to really think in mind, in a context of AIT, particularly when we are considering the effects of immunotherapy. We know that it requires up to 3 years, is associated with reduction of symptoms and the need of rescue medications. Also restored and supports the patients for not having to have a loss of productivity, and function particularly. And of course, there's a reduction, there's an element of prevention here, where when there's a likelihood of developing additional allergies, and also particularly new sensitization. And there's an element of long-term sustained responsiveness. So there is data that supports that you give immunotherapy for 3 years, you can have clinical efficacy for up to 5 years, and even more importantly, the fact that you have sustained responsiveness, sustained clinical responsiveness of treatment beyond discontinuation of treatment. And here the question then is, just to heat up a little bit in terms of the Q&A and the question, which immunological mechanisms best explains the long-term clinical benefits and sustained tolerance observed after completion of immunotherapy? So what are the mechanisms of AIT, particularly when we think about long-term benefits? Again, I'll hand over to Christian.
Thank you very much. So you can choose A, if you think it's a temporary desensitization of mast cells and basophils leading to reduced histamine release in the early weeks of treatment. Or if you think it's B, the initial reduction in eosinophil recruitment during intermediate phase of therapy, then choose this. C is the shift from Th2 to Th1 immune response that occurs within months of starting treatment. Then we have D, establishment of durable allergen-specific regulatory T cells, also known as Tregs, and the persistent IgG4 blocking antibodies that maintain long-term immune tolerance. Or you can also choose E if you think that's correct. It's the increased activation of effector T cells, enhancing the immune responsiveness after therapy. And I see a lot of answers incoming. And again, to speed us up, I will start the countdown timer, giving you the last 5 seconds to vote. All right, so let's have a look at the results, and you can clearly see that more than 80% have chosen D, the establishment of durable allergen-specific T cells and persistent IgG blocking antibodies. What do you think about these results, Mo?
Well, fantastic. We have we have an audience that knows very much about AIT and mechanisms as well, from diagnostic to patient management to the mechanisms of how it works. So very pleased. And the answer, of course, is D. So thank you for all of you who have voted. And at least now we know that you need induction of regulatory T cell responses. We need blocking antibodies. We need several, and these somewhat contribute to the efficacy of treatment. Right. So one of the things that I found very important, and I, over my career developments from a junior fellow, I did a training as a clinical scientist in immunology at Imperial and Chelsea Westminster Hospital, and then did a PhD, and then developed my group, build it up, and we have one of the important department that really focuses on immunotherapy, and I've learned certain key lessons that I would really like to share with you. And I do get lots of fellows who come to my group, and they are this, and we really discuss, and career development is very important, and success in research is also very important, because it is tough, but one has to be resilient. And I think one of the key elements for me in terms of which got me to where I am today is certainly resilience and ethics of how I work. And one of the other important aspects that has contributed to the success, I would say, is being able to be adaptive. Adaptive to setbacks. We know that when we do experiments, certainly when I was doing my PhD and doing experiments in the lab, and 60 to 70% of the experiments will be negative and exploratory. But I think that really helped me to be to try to be very motivated and be resilient and just push forward and forward. And I think eventually one does break through this sort of barrier, and I think that's extremely important. So it's important to really try to be very adaptable and really appreciate that there will always be setbacks, but the key is how do we break the roof, and how do we carry on going? And I think that's really important to think about that. It's extremely important to remain motivated. One, research can be long. Some of the studies we work on takes 5 years, and the first 3 years is just working with clinical colleagues and being able to collect samples and perform biobanking, and then the creativity starts. But I think resilience, persistence, and really trying to think about how best and just look forward. And I think that has been some of the key elements that have been very important in terms of also thinking about setting goals, realistic goals, short-term goals. Can also help you really move forward and remain motivated. So rather than long-term goals, you have several short-term goals, and then you those are the cumulative of those can really support. And also thinking about collaboratively working with others and see how they survive in the field of research or translational research, and just sharing perspectives and also when things don't go well, it's always good to talk about it too. It is important to think about self-care and having the relevant support system. So I think it's important, as I mentioned, not all experiments work, and I think it's good to have some very good peer system. Peers are usually good. We can have fellows that you are able to talk to, mentors particularly, and also friends. I think mentors are there to really support you through your career development. And I think it's really important to think about building a relationship. I think once you've done 1, 2, and 3, is then you're on an upward track where you then have to build relationship and maintain them to be successful. One thing is building one, and the second thing is to maintaining it, and the third thing is to get the most out of professional relationships. And I think it's really important to think about mutual respect, support, considering knowledge sharing. How can one work together? The era of I do all by myself and I publish all by myself is gone. I think we are increasingly having to work as a team science, and I think that's really important. And the way to have a successful team science is to really think about working and collaborating as a collective. If you look at the studies that have been published in the high impact journals always have multiple teams. And of course, as part of career development, we always think about first and last authorship, and the quality of the study, the impact of the study. Of course, that's important, but the first and the last is very important in a way that one can consider in terms of career promotion. You need as many first and as many last. And also, it's also very important to also think about what type of articles you'd like to author. Original articles are much more favored. They show rigor of science. Reviews are great, but they're just reviews, they're peer reviews, but they're very important as well. Consensus documents are important. Guidelines are extremely important for our clinical colleagues and residents and so on. So it's very important to think about what you do. But at the core of it is a collaboration that comes in. Inclusivity is extremely important. Thinking about working with fellows, considering the particularly a cohesive approach to working together. Inclusivity, working together, you bring the best minds, and if you're amongst the best minds, you will progress very well, very promptly. So very important to think about collaboration. And of course, transparency is one of the key elements in terms of collaboration. Clear communications, transparency in if you have a project, you want to collaborate, you know that it's extremely important to collaborate. It's also very important to discuss the output and how this will be this will develop at the end of the project. It's just demonstrating vision. It's thinking about how best to work together and to appreciate that everybody needs career development and how this could be accommodated. And what's really great in terms of the teen science today, and if you look at the best studies that have been published, you have 1 or 2 or 3 first authors, and also you have 1 or 2 or 3 senior authors. And that means that you're bringing people together. And then it's a way of working very well together in a transparent way with mutual respect, and particularly with a good communication relationship. And I think once you have that, then you have to then, that's just the beginning, and then you have to maintain it, because, you may want to do several other studies as well in the future. So those are my key sort of 6 points that I thought I'll share with you today, that at least has helped me along the way, in the way that I think about research and collaboration with my clinical colleagues, with the data scientists that I collaborate with, and with everyone that I work with.
Thank you so much, Mo, for sharing, first of all, your case. I didn't see any questions about that. So it's good that you already transitioned to the career part, which we always like to talk about. And for your honest feedback or do's and research, we could basically say. We want to discuss that a bit as well. The first question, I think, comes from Anne-Marie.
Yeah, I was very interested in asking you to try and look back and think about which career decision was most pivotal in shaping your trajectory that you have gone upon in your career.
Well, certainly what really helped me most was the decision to do a PhD. Although my PhD was in clinical medicine, I'd actually spent in an allergy lab. I worked with Stephen Durham, and I was exposed to translational research. And that really shaped me in a way that I think, in a way that I was exposed to research, in a way that then, you are in an environment that is very supportive. And then you have to have your own, you have to then build your own path amongst that, right? And as you build your own path, you take the best that you were exposed to, and you try to work within that framework. And develop that, and sometimes it works and sometimes it doesn't, but you break through. It's like always breaking the ceiling, and then you see what works is what you maintain, and then you really learn over the years. Budding out as from a PhD to a first investigator to then having your first group, the excitement of having a student when you have your doctorate is great, because you feel like, oh goodness me, like now I have a student. I was one, and then you have 2, and then you have to learn a bit of management, right? Yeah, and then the management skill comes into it, and then you start thinking, okay, well, the vision is this is what I want to do. So you build a group, then you think, okay, now I need collaboration, and I need sustainable collaboration for funding and so on. And that was really the key seminal point for me that really put me, because I really enjoyed translational research. I felt that I wanted to make an impact, and I felt that the best way to do from bench to bedside is to do, to think about allergy as a specialty, because it's achievable. And secondly, collaborating with clinical colleagues is even more important. And then the contribution I can make in a context of understanding mechanism, in a context of identifying relevant biomarkers and so on, and supporting some of the work from bench to bedside going into the clinic. So that really helped me, the first experience. And I could say doing a PhD in the right place at the right time, I think for me was the best decision I had made.
Right, thank you.
We talked about the do's, but I also want to talk about the don'ts. We always try to learn from mistakes. So what was your biggest mistake that you made, or the biggest regret that you have about your career choices, or something that went horribly wrong, and also something that you see many people make as a mistake?
Right. It's difficult, isn't it? We all have different experiences. And I wouldn't really say there's something wrong. I think what I'd say is I've learned from some of the decisions I made along the way. I think it's important to appreciate collaboration. It's also important to consider, as you, one has to also appreciate the limitations in the work they can achieve. So it's great to be, to think about moving ahead and having a clear vision and pushing forward. But somewhere along the line, it's important to think, okay, well, let me take a step back. I can still achieve what I want to do, but I need to be careful on how I am doing it, because there is an element of having a whole team behind you for the mission that you have, and it's important to just think that you can't make mistakes, and therefore you just have to just think and step back, and that's really that's what I can share, really. I think there are no wrong or right, or the do's and the don'ts. Of course, there's a lot of the do's. The don'ts is they are like, I think it's important to appreciate collaboration. So, just the fact that you have a project doesn't mean it's yours. So it's a project for everybody, and everybody contributes, and it's really important. So in my group, we have a team, and everybody contributes, and everybody needs to feel like they contribute, because if they own it, it's even better than just me saying that it's a project, it's our project. It's not my project, it's their project. And I think that's really the key for me. And I think it's also very important to think about when you're collaborating, and this is not from personal experience, but I think it's just general, don't replicate who you want to collaborate with. I think that's very important, because it's complementarity. That's why they are collaborators. Collaborators are those who want to collaborate with you because they bring their strength into the group. So reinventing the wheel doesn't really help, because it just sets you back 5 years or 10 years anyway. So, I think it's much more fun to collaborate and keep going.
Thank you for being so open also about possible mistakes that could be made. In the last few minutes of our webinars, we always try to torture our guests a little bit with some rapid fire questions. We ask you to answer them in just a few words, or just a single word if it's an either or question. And we're going to alternate asking questions for you. And I'm going to get started right away. Biomarkers or symptoms, what should drive future AIT decisions?
I think it's a combination of both.
You can answer that.
Oh, yeah, you can.
Of course, I would say biomarkers, right? With AI, naturally.
Right. Okay. Machine learning or classical statistics, which will dominate allergy research?
I think we'll see increasingly AI coming into the play.
Yeah.
If you had to choose again, become a doctor or a researcher?
I'll do it again, researcher for sure.
So multi-omics or single biomarkers, where is the future of precision AIT?
Multi-omics with composite biomarkers.
Early life prevention or adult disease modification, where do you see more possible impact for patients?
Oh, early life prevention for sure. I think this is what we really need to strive for. We have to strive for that.
Yeah. And now it gets a little more personal. When everything gets too much, what is your personal reset?
Classical music.
Data privacy or data interoperability, what is the bigger challenge?
I think the latter.
Scientific journal or leisure book?
Scientific journal.
And the last one, do you have any allergies yourself?
Oh, I do. Yes. Houses, smiles, and grass.
Amazing.
Well, I think you got someone who can treat you well, probably. So thank you for enduring our rapid fire questions. We're at the end of our webinar, and I first of all want to thank you for the presentation and for Anne-Marie for hosting with me, our audience for taking part in all the polls. And at last, we want to give back the word to our moderator, Christian Schwager.
Yes, thank you very much. First of all, I want to say sorry because we have noticed that some of our translation did not work that well. We will promise we keep working on it, including more languages and somehow optimizing the whole translation stuff. So in the next webinar, it will be much better than this time. And so first of all, or second one, I want to thank Professor Shamji, Mo, who presented with great enthusiasm and showed that AI can really contribute to allergen immunotherapy. And then, of course, I would like to thank Anne-Marie Schuss and Sebastian Zurich, who have guided us through this evening with confidence, clear structure, and just the right amount of charm. So really thank you from the bottom of my heart. And the last thanks goes out to ALK, our sponsor, and with the continuous support of ALK, we are able to offer this great webinars. And if you like our webinars and are eager for more Fascination Allergology, we warmly invite you to our next webinar, which is scheduled 11th of March, featuring another distinguished expert, Professor Oliver Farr, who will illuminate the topic of evidence in allergology. And to reduce, of course, the language barriers, we will implement the AI translation again, hopefully with a bit better effect than this time. And please stay with us for a moment. After the brief feedback survey, which will start soon, you will be redirected to the registration page of the next webinar, so you can just register in a few moments. And I want to thank again Mo Shamji, my two co-moderators, and we are looking forward to seeing you next time when it's time for Fascination Allergology. Until then, we wish you all the best and a pleasant time ahead. Thank you very much.
Bye-bye.
Pleasure.
Bye. Thank you.
Mhm.